Hello friends, and welcome to a video lecture on topic A3.2 Classification and Cladistics. Our guiding questions for today: what tools are used to classify organisms into taxonomic groups, and how do cladistic methods differ from traditional taxonomic methods? Our objectives: we're going to talk about the need for classification of living things. We're going to define taxonomy. We're going to explain the bases for classifying organisms into different taxa. We're going to analyze some cladograms and then review those three domains of life. So this idea of classification, we're placing organisms into groups based on traits and/or evolutionary origins. But why do we need to classify living things? It's because there are a lot of living things. And so there's a lot of information regarding them. We have needs to store and retrieve and apply all that information. There are an estimated 8.7 million different species on the planet, and that doesn't even include all the different kinds of bacteria. We have classified 2.15 billion of them. Most of them are insects. We've got some spiders and other stuff, us mammals. There aren't so many of us. But because there are so many different organisms out there, we need to classify so that it is easy for us to communicate with each other about these organisms. We place the organisms into different groups. These groups are known as taxa. Taxon is singular, taxa is plural. They are classificatory groups. Taxonomy is the process of assigning organisms to those groups. We have a hierarchy of taxa shown here. Domain is the broadest, the most inclusive of our different taxa. And then we have kingdom, phylum, class, order, family, genus, species. You can remember this because dumb kids playing cards on freeways get squished. This is our mnemonic device for the hierarchy of taxa. Dumb kids playing cards on freeway get squished. Domain, kingdom, phylum, class, order, family, genus, and species. And of course, genus and species is going to give us our binomial nomenclature, the scientific name of organisms. What can be a little bit tricky about classification is that it is very much a human construct. Us humans decide what organisms go into what groups. And sometimes, especially during speciation events, it's hard for us to know exactly when a group is now two different species instead of a single species. This is known as the boundary paradox, at which point do we say, haha, they are actually different species because they're super different and they can no longer interbreed. But what about in this little bit of time when maybe they could interbreed and produce fertile offspring? It's a little bit tricky to know exactly when those speciation events occurred. It can be tricky to know when these different groups are actually different groups and not just the same groups with variation. Because this is a human construct, sometimes us humans disagree on classification, and this has been a little bit of a struggle for us historically. Luckily, technology has advanced, and it's allowing us to classify organisms based on evolutionary relationships instead of just morphology. We definitely still use morphology to help us classify organisms. Morphology is the study of the shape or form of organisms. But more often we're using DNA sequencing and the amino acid sequences of proteins and looking at the similarities of those sequences to determine how closely different groups are related to each other in terms of evolution. We've actually reclassified quite a few organisms based on their DNA sequences, their amino acid sequences, because sometimes we have those super cool analogous structures that are formed through convergent evolution that make organisms look alike, even though they're not actually that closely related to each other. We can use this information, making sure that we're following these two tenets of classification, that all the organisms evolved from a common ancestor are included in a group, and only organisms evolved from that common ancestor are included in a group. This diagram is a cladogram, and it helps us illustrate the evolutionary relationships amongst different groups of organisms. Here I'm showing two organisms that have a common ancestor, and that common ancestor was here. I'm going to call that a node. That node here is going to this particular node ending in terminal branches, because I don't have any more branches after these ones. These terminal branches are showing us groups of organisms that have a common ancestor. There it is. I did not include this organism over here because it doesn't have that same common ancestor. Instead, this is the common ancestor shared amongst those three organisms. We're just going to follow the lines back in time until they run into each other. So this node is showing us the common ancestor of all of these organisms. Everything that comes after that point in the cladogram is going to be part of a clade. It is going to have common ancestry. But again, our cladograms, these are human constructs. The more technology we build, the better our cladograms become. But know that sometimes we're going to have to change our cladograms. This cladogram is pretty cool because it includes some morphologies that we're going to call synapomorphies. Syn meaning together or at the same time, and morph meaning shape or form. These are shapes or forms that are shared by all of the organisms after that point in the cladogram. So all of these groups have a preoral tentacle, but only these groups are going to have dorsoventrally elongated bodies. Here's another example of a cladogram. This one's pretty cool because we are now including some extinct species. We still have our nodes. These are those points of divergence when we had a common ancestor here that went through some kind of speciation event. Now we have multiple species from that one common ancestor. It also is going to show us clades. Clades are pretty cool. They are groups of organisms that share a common ancestor. We can have lots of different levels of clade. This is a clade, and these two guys share this common ancestor, but also this is a clade, and they share this common ancestor. We can actually call this one a nested clade because it's nested within this bigger clade. But then, of course, I could also talk about this clade. And now this whole thing is also nested within that one. So again, lots of levels of clades, lots of levels of nesting. These ending branches, the ones that don't have any more divergent points, these are known again as terminal. They are terminal clades. And what's super cool about this particular cladogram is that it includes extinct species. So I'm including all of the species that evolved from this common ancestor. I'm going to call this the root because this is the beginning of all of the descendants. This root, this common ancestor, gave rise to all of these different species of, these happen to be bears. But what's super cool is that only the green fonted bear species are the ones that are still in existence. These guys, all extinct. And so we absolutely can include all, even the extinct species that are evolved from a common ancestor. It can be tricky for us to include these extinct species because, of course, it's really hard to analyze their DNA when they're extinct. This particular cladogram is super cool because it doesn't have organisms. It actually is showing us the evolutionary relationships amongst different molecules of hemoglobin and myoglobin. And what I love about this particular cladogram is that it includes the number of DNA differences that have accumulated over time from those divergent points. So this ancestral molecule that went through some mutations and diverged into different molecules has 257 different DNA sequences. And what is super cool is that we know through lots and lots of research that there's just about 10 to the minus 9 mutations accumulating in DNA per year. So if we see 257 DNA differences, 257 mutations between myoglobin and this ancestral molecule, then I can do some super easy conversions. 10 to the minus 9 mutations per every year is going to lead us to 257 times 10 to the 11th years when I divide 257 by 10 to the minus 9, cancel out mutations, and we're left with years. So 2.57 times 10 to the 11th years ago is when we had this diverging event. We could rewrite our scientific notation into, that's going to turn into 257 billion years ago. We can call this particular kind of cladogram molecular clock, because not only is it showing us relationships amongst the different molecules, but we're also being able to see how long ago, what kind of time is involved in those divergent events. And we build these molecular clocks by looking at similarities amongst DNA sequences and amino acid sequences for proteins that are present in lots of different kinds of organisms. This is the amino acid sequence for the protein known as cytochrome C. It's involved in respiration. And here we have for yeast and humans and horses and mice. And all of these different letters are just showing us representing different amino acids. And we can see that there are quite a few places where our amino acid sequence is almost the same. So we're keeping all the H's and all the T's and all the K's. So lots of similarities in lots of places. There are a couple spots where we see quite a bit of divergence. The more similar the amino acid sequence, the more closely related in terms of evolution are the organisms. And what's super cool is that scientists, organizations have published databases that anyone can access to look at DNA sequences, protein amino acid sequences. This one happens to be the European Molecular Biology Laboratory, European Bioinformatics Institute, and you can find a gene or a protein and then look at its sequence, which is super cool. And we're going to wrap up this video lecture with a review of something that we talked about at the very beginning, the fact that dumb kids playing cards on freeways get squished. Domain, kingdom, phylum, class, order, family, genus, species. And again, genus, species makes up binomial nomenclature. It is a scientific name of an organism. We are going to capitalize genus, lowercase species, and the whole thing is going to be italicized. What's crazy is that I'm kind of old. And so when I was in school, we really only talked about kingdom, phylum on down the list. We didn't really talk about domains. We talked about two kinds of cells. There were eukaryotes and prokaryotes, and you either had a nucleus or you didn't. But then scientists started to look at rRNA sequences. This is ribosomal RNA. And scientists figured out that prokaryotes are not all the same. And there were so many differences in those rRNA sequences that scientists decided that we had to change our classification system and have not just prokaryotes and eukaryotes, but we needed actually 3 domains of life: bacteria, archaea, and eukarya. These are 3 domains of life now, and this classification change was made because we had better technology and we could look at ribosomal RNA. Bacteria have naked DNA, except archaea, they do have some histone proteins. What madness! Eukaryotes have introns and bacteria don't, except sometimes archaea do. What madness! It's like everything that we thought we knew, we don't know anymore. Because of these major differences, scientists decided that what we thought we knew, we didn't know, and we had to make a change. How super cool is science that it can reflect upon itself and learn and grow and know more. Again, we used to have only 2 big groupings, prokaryotes and eukaryotes. Here's our common ancestor. This happens to be Luca, our last universal common ancestor. 1, 2, 3 domains of life, this beautiful cladogram, which we sometimes call the tree of life. And can you believe, under 15 minutes and we have wrapped up this video lecture already. We talked about the tools that we use to classify organisms into taxonomic groups. Sometimes we look at morphology. More often we're looking at DNA, RNA, and protein amino acid sequences. How do cladistic methods differ from traditional taxonomic methods? We are using that molecular biology in order to look at evolutionary relationships instead of just morphological similarities. We talked about the need for classification. There are so many different kinds of living things. We talked about taxonomy, classifying organisms. We talked about how we do it. We can use morphology, the shape, the form, how an organism looks, but we can also use that molecular biology, DNA sequences, RNA sequences, and amino acid sequences. We analyzed our super cool cladograms, and we talked about the 3 domains of life: bacteria, archaea, and eukarya. Great work today.